Our next speaker is Art Toga. Like Richard Gibbs and Gregor Eikela, when we were starting the Atlas project, Art was a great advisor. I think he was actually at the early planning charrettes as well. And like Richard running a genome center, Art was running a brain mapping center. And the expertise that he brought to our project in the early stages for understanding how to map into the brain framework and having the IT infrastructure to support the work that we were doing was incredibly valuable. And it's a great honor to have Art join us today. Art is now, he gave us all of this help when he was at Lonnie at UCLA, and he's moved all the way across LA to USC. So welcome, Art.
Thanks. Moving across Los Angeles is a lot farther than moving across the country, I can tell you that. And the other thing about the charrette, while he's setting us up, when I was asked, and my story is very similar to Joe's. Let me see if I can get this to go on while there we go. When I was asked to participate in a charrette, I had no idea what the word charrette means. I'm not sure. Does anybody in the audience not know what the word charrette means? Thank you. Okay, so I didn't, I'm not quite as stupid as I thought I might have been. So if you look it up, which I had to at the time, it means, I got nothing here, by the way. You've broken my machine somehow. Wait a minute.
Let me unplug this.
We're good. There we go. So if you look up the word charrette, it has something to do with a French cart, which was used to haul away the students after they had to work very hard under a very tight deadline, because apparently they'd be exhausted and couldn't do anything else. So, in fact, the charrette was a little bit like that. So, Paul, that was a remarkable experience. But thank you for that. I came home very exhausted. My wife said, "Where did you go?" I said, "I can't tell anything about it. It's all secret." So anyway, let's see if I can. It wants to restart. Don't send. All right. Let's see what we do here. Okay. I think we're good. No audio. No. Just my audio. Can you see on the screen there? Yep. No. Okay. Sorry for that. So we're going to change directions a little bit. We're going to move down from the very small area looking at genes and molecules to looking at whole brain. And I think one of the points that I'm going to try and make in my 20 minutes or so is that examining the phenotype relating to the genotypes that we're all interested in here is important in many respects. And phenotype can come in a variety of ways. And we should not necessarily narrow our focus into a single measure of phenotype. That in fact, there are many ways to look at phenotypes, and they all have varying degrees of association with the genotypes that are being studied. And I think if you look at a slide like this, you can readily appreciate that there's really quite a variety in scale that can be examined phenotypically as one looks at the whole brain or the organ system itself down to molecules. And all of these things have a role to play. And the second message I'd like to deliver in my 20 minutes has to do with the integration of these different data types and different scales, and that we should always try to incorporate as much of this as possible, because in fact, this is a complex organ system. Its structure and function can be examined in so many different ways, using so many different modalities, and it's really up to us to take all of these perspectives to get a comprehensive whole. And that's easily said and difficult to do, as everybody in this room recognizes. And in my career, I've spent a great deal of it trying to utilize imaging technologies, and there are many, to witness both structure and function in vivo, in humans, as well as in animal models. And this slide that you probably can't appreciate is a neurosurgical view of an open craniotomy patient. And this is what a surgeon sees. I don't know if there are any surgeons in the room, but trying to relate what it is that we can see, witness, and measure in an experimental situation relative to a clinical case is extraordinarily difficult, and one that I think shows really the complexity of the brain. This is really what it is when trying to make measurements from here. And we're trying to determine what it is these images are saying about the brain. So I started this over 30 years ago and witnessed the onslaught of MR, which is now ubiquitous in a variety of studies, as you've already heard today. But a lot of what we had to do is we would look at images such as this and try to improve the signal to noise and relate it to what we could see microscopically. This is actually the average of 27 different scans of the same postdoctoral fellow. And we did that just to improve the signal to noise so that we could perhaps get down to the laminar distinctions of certain nuclei. But more importantly, we wanted to be able to appreciate what it is we could see in vivo in a living postdoctoral fellow with this post-mortem. It's not the same postdoctoral fellow. But it gives us a relationship between what we can see at the cellular using light microscopy relative to what we can see in vivo using MR. And at this resolution, you can't see a lot, but of course, because it's histologically prepared, you can then take these sections, scan them up using a light microscope or even any other way, reassemble them, reconstruct them, and then relate what it is that you can see really at a 39,000 foot level, because you only see in the millimeter or so resolution, to what we can see microscopically, even down to single cells. And of course, it generates massive amounts of data. So one of the other things that I think all of us have been able to experience is the amount of data that's generated is ever increasing, and we have to employ very sophisticated computational strategies to develop methods to handle these data and make quantitative measures. The second thing you can do with these tissue sections is then look at other architectural features. Much of what we've done here is looking at anatomical features that you can see, and they've been historically derived for over 100 years. But now we can subdivide regions of the cortex, not only in the genoarchitecture, as you've just heard, but looking at chemoarchitecture, looking at receptor patterns relative to the features that might be able to be witnessed in cell packing density or other characteristics that can be seen light microscopically. I'm going to diverge for 1 minute and tell you a funny story about this. These are post-mortem slab sections of humans, and clearly the Allen Institute has been involved in this as well, so you know the difficulty. Well, we were doing this years ago with a colleague, George Paxinos, who's an anatomist from Sydney, Australia. And he did a sabbatical in the lab. And at the end of the sabbatical, he wanted to take some of these tissue samples with him so that he could continue the work in Sydney. This was before 9/11. So we decided, well, let's just give you the whole specimen, and you can slice it up as you want. And as you saw in the slide that I showed, the brain was still in its container. It was still in the cranium. So we took a person's head, and we put it in a cooler, styrofoam cooler, and filled it with dry ice. And we got all the paperwork for the biohazard tissue. And we sent George down to LAX, and I went with him to put him on the airplane because it's only a 14-hour flight after all, and the specimen would be frozen, no problem. What we forgot to realize, of course, is you put the cooler in the X-ray machine. So we put the cooler in the X-ray machine, and I'm waiting for George to, okay, George, we'll see you. And the guy's watching the monitor, and he sees this, and he goes, whoa, 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 what is going on here? And of course, you could see a human head in the cooler. And it was embarrassing, and we got through it all. And of course, the joke of the story, this is an absolutely true story, but the joke of the story is, where do you put it when you get on the airplane? Come on, you put it in the overhead compartment, sorry. Okay. Okay, so the other thing that you can do is you can go down to animal models, of course, and use the same kind of MR techniques and relate those with what we can see post-mortem using histology. And this was a lot of the work that we were doing when this effort 10 years ago began. And so we started some of these things in terms of finding atlases or creating atlases that had stereotactic coordinates associated with them in 3D. Because you may make sections that weren't quite in the orthogonal plane of whatever normal atlas you might use. And some of these things provided a computational strategy so that you could make morphological measures between different animals. And more importantly, we could do MR on the very same animals. That's not what I wanted to do. I just went backwards. Okay. Sorry. That's what I want. So you can do MR on the very same animal. This is a mouse. We took the same MR animal. We then physically sectioned it, reconstructed it using a block face approach, which is one of the ones that's used here, and then took the tissue sections, all the while keeping track of where we were. This was done in concert with Marianne Martone in San Diego, some of you know. We scanned up the tissue sections, remember keeping track of where we were, zoomed down into individual cells, scanned those using high voltage electron microscopy, and then keeping track of where we were, modeled the processes on the end here, looking at synaptic boutons and other features, looking at them not only in terms of the raw data, but then modeling these using a graphical approach. This is where we're headed. I actually believe, maybe not in my lifetime, but in some of yours, we'll be able to do this in a comprehensive way. By reducing the field of view to a way where we can look down at individual cell processes, we can get exquisite detail from nanometer all the way up to centimeter. We just can't do it in the whole brain yet, but we will. And I think that's an important thing to try to strive for, and perhaps the Obama Brains Initiative will provide the resources to do something like that. Now, in the animal model, it's easy to do, because there's not so much variability. They're relatively genetically homogeneous. In the human, that's not the case. So in my world, when we're trying to look at cohorts of normals or disease groups, we have to deal with the extreme variability in individuals. This is 10 people, sagittal sections, looking at the corpus callosum. There should be a pattern obvious to all of you here. And everybody sees that pattern, right? Some do. Not a lot of nods. This slide comes from Mike Gazzaniga from a long, long time ago. He was dealing with interhemispheric issues, but that's irrelevant here. Just look at the morphological similarity between the pairs versus the columns. So they're far more similar looking at each of these, and that's because those are 5 pairs of monozygotic twins. That tells you 2 things. One is genes play a role in some crude morphological similarity, such as the corpus callosum, but 2, that environmental, experiential, and other factors continue to modify the shape, form, and position of this particular structure, even though these folks are monozygotic twins. And I think that's a very important thing, because as you're trying to do genotype-phenotype comparisons, it's clear that other factors play a significant role. We did lots of work in the mouse, and again, this is back about 10 years ago, using some of these older atlases to relate these different data types. And I just wanted to show you this slide in comparison to the human, because you can obviously do destructive sampling more easily in a mouse, and you can relate the various types of measurements from genes to histology to MR, all being able to put these things back together again. The variability of the human, as I said, does present some unique challenges. You have to develop some fairly sophisticated mathematics. The people here at the Allen Brain Institute obviously have solved that in the mouse, as you can look on the website and see the relationship between all those animals. In the human, it's a little more difficult. The features are more complex. You have to employ this math, which I'm going to spend the rest of my time going over the derivation of this mathematics. I won't do that. Instead, I'll show you a very quick movie. And again, these are the kinds of very active literature that is important to incorporate the expertise of other disciplines. Most of you here are neurobiologists of one type or another. This is done with mathematicians, physicists, computer scientists, and I firmly believe that having a team that includes the expertise of all these people is essential to creating a successful program in order to solve some of these very, very difficult problems. And this is just a series of images that are showing you the drive of a warp to relate one brain relative to another. Of course, you could do it from an individual to an atlas, and all of that allows you to understand the variability. By making things the same, you've mathematically determined exactly how different they were. In our case, in the human world, we deal in a probabilistic sense. There's no absolutes because everybody's so variable, and so we want to probabilistically know where we are, what features are there, what kinds of measurements we can find. And we have to accommodate that variability, because if you think about it like an analysis of variance, does this person belong to this cohort, or are they in this disease cohort? And again, this just shows you the degree of difference between 2 individuals, both normal adults, just showing you the degree of distortion that must be imposed to make them equivalent in space. And again, this goes from a planar representation to spherical representation and then back into its native space. It's computationally expensive when you're dealing with thousands of subjects, which we do, but I think it's an exercise that is essential to understanding what that population is representing. And again, you can just kind of see the degree of shearing and deformation necessary here. I'd like to use as an example the application of this, of a particular disease that threatens us all. If you live long enough, you have a high likelihood of getting Alzheimer's disease. I think Viviana, you said this, right? So she was talking about Parkinson's disease, but there are a number of neurodegenerative diseases as we get older that we are increasingly at risk for just by a function of our age, notwithstanding any genetic risk we may have. And it's important to make as much progress on this as we can, because it's a very expensive disease for society. These are maps of Alzheimer's disease. You know that there are susceptible regions of the brain, more so than other regions, limbic structures, temporal lobe in particular. But if you look at this little movie here, this is a single individual. And again, this Gregor Eichle might appreciate this, looking at the swelling of the ventricles. That was a very interesting talk this morning. This is what's going on in this disease process. It might be interesting to look at some of this these cilia patterns in these aged individuals. 4 years apart. That's how much tissue loss occurs in that individual. If we make a movie between a normal individual and an AD individual, again using the deformation strategies I just showed you, you can appreciate the devastation that this disease imposes on the cortex itself. It erodes a vast amount of tissue. Now the question in a clinical sense is, we know this. By the time you can see this using MR, the patient has clearly got Alzheimer's disease. They're suffering from cognitive losses already. This does us no diagnostic utility whatsoever. More importantly, we want to be able to move early in the progression of the disease in the prodromal state so that we can employ various therapies as they come online. This is a probabilistic representation. I keep doing that. Never mind. There we go. And this one right here is, this is an average of about 17 subjects, I think. And these people, you can see over a course of 2 years, the progression of the gray matter loss that originates in this region of the brain, moves out from limbic structures. This is an exterior surface view, and in a minute you'll see a midline view where you can see from limbic structures through cingulate and beyond. So it's a wave of tissue loss. And there's a reason that certain cells are susceptible versus other ones. Just to keep moving here. There's a couple of other methods that we can use in studying this disease, and what we're really interested in is these MCI. It stands for mild cognitive impairment. It's certainly one of the prodromal states of Alzheimer's disease, and by using this battery of imaging, which we do now, one can appreciate the relationship between what can be seen histologically up here with that which can be seen in vivo using everything from tracer-based approaches in PET to fractional anisotropy, such as Pat Kuhl talked about, to diffusion imaging, which he also talked about, and resting state fMRI. Again, these are multiple windows into what's going on in a holistic way as this disease process progresses. Diffusion imaging is really one of the more exciting aspects in the last 10 years, maybe not quite 10 years. This is a project called the Human Connectome Project. We are one of the awardees. There's another one, I think. I just can't remember the name of them. But anyway, that's an inside joke for those of you who don't know that. The notion here is to map in a comprehensive way the human connectome using diffusion imaging and then try to define the wiring diagram of the humans. This is a very interesting technique. It provides exquisite imagery. But I want to be clear. It's not the same as doing injections and finding fibers going from point A to point B. You cannot see that. All we can see is fibers. We don't know the actual connectivity. And so when you read these papers, read them carefully. But using high fields, as I'll show you here, one can appreciate, I don't know how well you can see it back there, but you can see the U fibers and the detail available using this technique. This is an extraordinarily promising technique, and at 7T, such as here, the value of this data is really quite amazing. And I think as we move forward, you'll see techniques, as we are developing and others, that allow us to combine the wiring diagram of an individual with the wiring diagram of populations to try to discern relationships between disease groups and undiseased groups. Clearly something like schizophrenia or even autism, these are connectopathies, that these are wiring diagram problems that affect the behavior of these individuals in a serious way. One also can relate these wiring diagrams or these connectomes with other measurements. We can use computational strategies so that the wiring diagrams provide us with the linkages between what terminates with what with the precision that we have. And provide these connectomes, if you will, that allow us to make comparisons and relating that to other observations such as gray matter volume, gray matter thickness, and other measures begins to give us a much more comprehensive picture of brain. And that's important to recognize that one has to combine these techniques to get a full view. And again, the notion of connectopathies is evident here. Now, these are normal subjects, but one can find differences in sex and heritability, network measures, small worldness, and other approaches, all of which are available using these kind of connectome approaches. Relating the anatomy, such as you can't see here all of a sudden, there you can. The anatomy, such as the diffusion imaging that I showed you, and now it's gone again. I have it, you don't. There it is. Is it a loose connection? It's tight here, by the way. I'll go back to my joke telling in a minute. Any idea? It's your end, I think, not this end. I'll keep going, see what happens. There we go. So this is work that I want to show more on Connectome. This is work done by Hongwei Dong. He's actually an alumnus of the Allen Brain Institute. And he now does similar things to what he was doing with you guys, but doing sort of a tessellated injection pattern in 3D by making very detailed injections in these mice, and then doing both retrograde and anterograde mapping and scanning them all up into, as you can see, the tessellations right here. You can kind of see the various boxes, mapping them all up into 3D and providing comprehensive maps of the connections between these regions of the brain. And he's doing this in an unbelievably labor-intensive way. Probably learned all that while he was here with you guys. But I think the complementarity of this animal model approach is important relative to what we can do in the humans. And the other value of something like this with other groups working together is they're using the same Allen Brain Atlas effort that was developed here, using that as the same reference space. And so comparisons between work that's being done here in Seattle and work that's being done elsewhere is made possible by warping all of the data into a common reference frame. And I think that's an important construct not only in animals but as well as in humans. This is a project I'm going to finish up giving you some examples for the next few minutes here, some examples in the clinical case. This is a project called the Alzheimer's Disease Neuroimaging Initiative. It's a project where all the data actually comes to us. This is now a worldwide effort. I think Stephen Friend gave you some hint about some of this. These data come from a couple thousand subjects who are scanned and measured in every manner that you can imagine, controls, MCIs, and AD patients. The reason I'm showing you this is a cooperative study. Anybody can have this data, very much like the philosophy that was adopted here. This is done with human data. The net result, I think, Jim, as you mentioned, is this all bullshit or not? No, because the net result is the value of the data is considerable. The number of publications that's derived, and now there's over 500 publications. There's just under 5,000 active investigators on this. There's over 54 countries involved, and in North America there are 58 acquisition sites. All of this has produced a wealth of information in terms of specific biomarkers that hint towards Alzheimer's disease, which are important for the discovery for targeted therapies. And this is a cooperative venture with pharmaceutical industry as well. We've been involved with a number of these kind of efforts that are global and sharing in nature. This is the Alzheimer's one. This is the Michael J. Fox one, which we also host and run for Parkinson's disease. This is Huntington's disease. And this one was normal subjects. We couldn't resist that acronym. It doesn't stand for Intercontinental Ballistic Missile. It stands for the International Consortium for Brain Mapping. And I just wanted to show you one slide about normals, because that was about normals. When you read a paper about normals, they never tell you what it means. They don't tell you what normals are. And in fact, when you're doing subtle studies of looking at, subtle differences in brain, you really need to know what normal subjects are. I gave this talk last week, or a similar talk, and I said, I'm not normal. Those of you that know me immediately recognize that to be true, but the point is, a normal subject probably shouldn't be on any medication. I take statins. They probably shouldn't wear glasses. They shouldn't be unconscious ever in their life. You got bonked on the head as a kid. And so all of these kinds of descriptors for normal subjects needs to be incorporated in studies, and that's very rarely the case. So as you go forward in your careers, those of you that are still students, I hope you pay attention to that. Final few slides has to do with a project called Enigma. This is actually headed by Paul Thompson in our group. This is a replication through collaboration. Over 200 scientists, 12 countries, 125 institutions, 26,000 subjects. This is imaging data and genetics. What we do in Enigma is we compute brain measures from these scans. We have GWAS data, and then we do meta-analysis by combining the effects to see the relationship between genotype and phenotype as measured. And here are a few examples of those. What affects brain tissue loss as we age? Here's an interesting statistic. Obese people, morbidly obese people, have 8% more brain atrophy locally than those who are not morbidly obese. Just measured in these 26,000 people. Exercise may in fact protect the brain more than any current Alzheimer's disease medication. Now, there aren't really any medications. There are some acetylcholinesterase inhibitors. The bapineuzumab studies didn't work, so there really aren't very good medications. But pretty interesting to know. If you're active, it in fact may reduce gray matter loss. Stress and high cortisol levels promote atrophy. Those of us that have to undergo that periodically might agree, but here are some data that proves that. Homocysteine levels promote atrophy. Perhaps vitamin B is a thing to say here. And here's an interesting gene, FTO. I'm sure many of you know about this gene. The relationship between FTO and tissue loss is quite clear. And those of us in the audience, about 50% of us have FTO. And we will eat about 200 calories more per day than those that don't have the gene. And if you have it, there's more specific tissue loss in the brain throughout life than if you don't. Just another interesting observation. Final little genotype-phenotype association I have has to do with CLUE, and this is not gray matter relationship, but low fiber integrity. And so the point I'm trying to make with this series of examples here is that the relationship between genotype and phenotype can be varied, and there are lots of ways to make these measurements. And I encourage all of you to think about a number of ways in which you can make these associations. I'm almost done, don't worry. So we need to have very comprehensive tools to deal with this. We're dealing literally with thousands of subjects. We're dealing with thousands of subjects, each of which may have dozens of scans with GWAS data or even whole genome sequence data. And so the amount of data and the amount of processing necessary to do this is difficult. If you want to give this data away and allow other people to gain access to it, I think that's an important philosophy. I agree with it wholeheartedly. You have to give them tools as well. This is one such tool set that allows people literally to drag and drop various glyphs around here. These tools can come from any manner of colleagues. There's large libraries of them. And this is a particular example of one where we wanted to say, if I give the system 2,000 subjects, can you distinguish which of you, all of you have MCI, all of the 2,000 subjects have mild cognitive impairment, which of those 2,000 will convert to Alzheimer's disease and which will not? Can you determine that by looking at all the data that we have? So we built this giant system, and we got an 87% accuracy rate. How do we know the results? Because we took it out of the ADNI study. We knew who already had converted, but we did it blindly. And I think these kinds of tools have enormous clinical utility, especially with the onslaught of new therapies as they need to be tested for their efficacy. So again, this is just sort of a joint analysis of trying to look at the phenotype as measured using a variety of imaging techniques and some of the genotype measures that we had available to us. So this is my summary slide. I wanted to, again, reiterate the fact that multimodal data that comes from different sources makes different types of measurements, can be related to genotype. All of these things, I think, provide a different window into a description of brain conformation, both structurally and functionally. And in combination, I think there's hope for us to find early in some of these neurodegenerative disease signals that allow us to prophylactically treat these subjects. Even if we don't cure them, we can slow down the progression of the disease. And I think that's a worthwhile endeavor. So with that, I'd like to stop. Thank you very much. Remember, I stand between you and the break, so very
Quick question. Hi, Art. Good to see you again. Since this is basically a different audience than last week at our neurotechnology conference in Aspen, I'll ask you the same question about Carl Dijsselroth's, the utility of his clarity process with your connectomics work.
Yeah, I mean, there are some people here that have more familiarity than we do. We sent a couple of people up there to Stanford well, down there from here, to work with him and to learn the technique. And so we have it in our lab. We're using it. It's involved with the mouse project. The depth, the focal depth of the tissue, even if you can get all the parameters right, probably doesn't exceed 5 or 6 millimeters at best. If you can get an objective that works that well. Human tissue obviously is considerably bigger, so it really doesn't lend itself easily to humans, at least in a more than a slab-based approach. But I think these techniques, and again, I think all of them have a significant role to play. I think the spatial integrity offered by clarity is enormously worthwhile. And in concert with some of the other more destructive approaches, by combining these in a way, you get these perspectives that I think will be really important. Thank you and good night.
Sorry.